So welcome. This is our first community meeting for this semester. And thank you so much for joining. I see a lot of participants. It's great. And today we have Daniel, who is a PhD student currently. We figured out just now that he is not going to be for a long time a PhD student. He's submitting, no stress though. So yeah, and he's a main developer of the LIANA and the pipelines and the methods. And today he's going to talk about LIANA and how to use it, I hope. And yeah, so thank you so much for coming and thank you for being our first talk. So if you share your slides, I can tell you if we can see them and then you can go.
Can you see them?
Yes, perfect.
Okay. Yeah, I mean, I'm a PhD student in the group of Julio Cesar Rodriguez. I am, as Michaela mentioned, towards the end of my PhD, and I've spent like the vast majority of it working on cell-cell communication. Yeah, my main project has been LIANA, and after that LIANA Plus, and I will present here basically LIANA Plus. Yeah, I focus a bit more on applications, but yeah, I think if there is certain things that we can discuss, I'm happy to also show like the things that I put in supplements, I guess. And yeah, so as I said, I come from the lab of Julio Cesar Rodriguez, and we're a purely computational lab. And as any computational lab, what we do is like we do stats, right? And then we find associations between things. And I think the twist is that we usually do it with some prior knowledge. And I think here the idea is that, yeah, it makes it a bit more interpretable. And yeah, that's like the main motivation, right? So when we think about LIANA is not an exception to this, right? LIANA is basically always prior knowledge driven. Essentially, like we have a lot of methods, like lots of methods that have been re-implemented, and I've tried my best to make it as flexible as possible. And there is like quite some prior knowledge behind LIANA, and the idea is to generate basically hypotheses for social communication, right? I will start with like a simple example, but I think this one is more focused to show the flexibility of LIANA. So there was recently a publication on metabolite transcriptome spatial results data from Ludenberg from Sweden. It's a paper that was recently published, Visari et al. In Nature Biotech. And it's quite cool, the data, since you have metabolite peaks, which are measured with MALDI-MS, MSI, and then you have also, you know, the standard Visium, and they actually do the sequencing and the mass spec on the same slides. So it's quite cool, the data. I think one challenge that I'm trying to represent here is that you have like different observations between the two matrices. So essentially you have like two completely different undatas with like different observations. And LIANA deals with this. I will not go into details, but yeah, basically they have this, one of the experiments that they do is that they have these mice and one of the hemispheres is perturbed, right? They put like basically an antagonist to dopamine, and this essentially lesions one of the hemispheres and destroys all of these dopaminergic neurons. So basically in one of the hemispheres, there's no dopamine, while the other ones remains intact, right? And we have 3 of those slides, and there is like some pathologist or biologist annotations of like regions, right? And recently, a master student in the lab basically worked to develop a metabolite protein or metabolite receptor focused database, and we're going to use this. And basically, in this case, I'll start with, so in LIANA, I re-implemented, we re-implemented a multi-view learning approach, which is MISTI, that was published previously in the lab, but now we did it in Python, and we made it a bit more flexible in the way that it works. And yeah, basically I use it to model metabolite peaks by 2 different views, which are cell type proportions inferred by basically the convolution, and also brain-specific receptors that we get from this metabolite database that I mentioned earlier. By the way, I kind of assumed that everyone has a more or less computational background, and I hope that's the case. If not, I'm happy to, like basically go back and explain more things in more details, but I'll try to also keep it a bit higher level so I don't go into too much details. But yeah, so basically we model these things, right? We have metabolites. We try to jointly see if receptors and cell types explain them to some part, and we see that, for example, in the intact hemispheres, we see that dopamine and this 3-MT, which is a metabolite of dopamine, are quite well explained by the receptors and metabolites, and this is basically what we expect, right? Since there is a perturbation of dopamine. And then when we look a bit more closely into dopamine, we see it's like relatively well explained, right? Like an R square of 0.5 is not too bad for what we're trying to do here. And what we also can do is that we can see which view contributes the most to these predictions. And we see that cell types are a bit like better predictors of metabolites in this case. And what is interesting is that when we look at the top 3 predictors, basically of the dopamine, we see that it's MSN2, 1 and 2. Basically, these are 2 cell states which are medium spike neurons, right? And they are basically the ones that secrete dopamine. So, like having a strong association intact while this association is gone in lesion makes sense, right? And then the third one that we see is this ERD2, which is like the canonical receptor of dopamine. It's quite simple, right? And then we can see that it's also different between intact and lesion hemispheres, right? And that's more or less it. So, but the thing is that here we're modeling interactions globally. So we do it for the whole slide, right? We get like 1 interaction per target predictor association or 1 stat. And yeah, basically another step is to also look at the regions in which these interactions are occurring. And we do this with like these kind of simple metrics, which are like basically spatially weighted similarity metrics like cosine, Pearson, or like co-clustering, like I think Morans, which were, I think some of those metrics were implemented basically in other frameworks in the past. I think here the idea is that they're a bit faster or equally as fast at least. And we also make it so that it's like kind of interchangeable across components. But the idea here is like, like you see where the dopamine intensity is the highest, you see where dopamine expression is the highest, and it tells you where does the interaction occur. And as expected, we can see that basically the interaction happens in the intact hemispheres, right? And it's also only in these 3 atom regions, which is basically where you expect to see dopamine. So that's nice. It's like a clean, simple example of like the flexibility, and that's more or less it. I think this is more of an introduction of what you have, like what I will try to present now. So I think here the idea is that, I mean, we're getting more samples and experimental design is getting more complex, right? And we're also getting data sets that you have spatial and single cell quite often, more often than before. And the idea is that LIANA can handle all of this, right? And we actually combine every component in LIANA in this data set that I will present. So it's a data set that was from Kraman Lab and our lab. It was published in 2022, and it's myocardial infarction, and we have single nuke, it's not single cell. We have single nuke samples following myocardial infarction, and it's from different regions of the heart, which can be fibrotic, which is basically fibrosis. Then you have ischemic, which is, let's say, like borderline dead regions, like super inflamed, and then you have myogenic, which you can think of as healthy, right? And we have more or less the same amount of samples between single cell and spatial data. So that's quite nice. And first, we would focus on the spatial data, and we'll go back to these basically local interactions, on top of which I basically do like a very simple non-negative matrix factorization to just find, let's say, patterns of coordinated ligand receptor interactions across the different regions of the heart, right? And what we see is actually what we kind of expect, right? We see that there's like an automatic elbow selection procedure that is implemented in LIANA, and it gives us 5 factors, quite arbitrary also, but they're quite nice in the sense that we see like factor 1 and 2 are, in 5 are basically enriched and ischemic. This is on the sample level, so slight level, and then we see that myogenic is enriched for factor 3, and factor 4 is enriched in fibrotic, right? So this is like log2 odds ratios. So they basically, if it's below 0, it would be negative, right? Below 1, it would be negative. And And then to make kind of sense of this, right? So here as a reminder, this is done on like only the ligand receptor interactions that we basically capture per each observation. And then on top of the loadings that we get, so ligand receptor loadings that we get from NMF, we do pathway enrichment just to make kind of sense of the data. And we see that these factors 1 and 2, which are the ischemic ones, have basically an enrichment of inflammatory ligand receptors, which makes sense. Factor 3, which is the myogenic or mostly muscle tissue factors, basically have a downregulation of immune genes or immune pathways. And then factors 4 and 5, which are respectively in ischemic and fibrotic, are basically enriched in TGF-beta signaling, and TGF-beta signaling is actually one of the canonical pathways that drives fibrosis, right? So the nice thing about NMF is that in addition to basically giving you those like soft clusters, it also gives you loadings for the features, right? So in this case, interactions, and we can see some interactions that we kind of expect to see. So these are interactions that have also been implicated previously with basically macrophage infiltration, and we see those are enriched in basically ischemic regions of the slides. So that's quite nice. And yeah, when you can visualize the factors, right, and you can see it's like I have to do some annotations here, but basically you can see it's like the ischemic region of the slide, and you can see that the interactions that are with high loadings are within that region. So that's very much expected, right? But the thing is that we find these interactions, and these interactions are actually not reported in the original paper, and they're quite interesting to see. And I will move on to dissociated data now. So again, like I think here you can think your standard cell phone DB, cell chat methods. Yeah, basically we have a ligand, receptor, a source, a target, and like some score that is assigned by the methods. And yeah, a source target, a source cell type expresses a ligand generally, and a target cell type expresses a receptor generally, right? This is how we would like to think about those. And we call them ligand receptor methods, I think, most generally. And yeah, I think there is like 9 of those re-implemented in LIANA, and along with this, we also provide the consensus of any combination of those. So it's quite flexible how you can do it. By default, it's only 5 of those methods. And I think here, I think the slide is a bit heavy, but all it shows is like a tensor factorization on ligand receptor loading that was introduced by Eric and Hatch, so Armingo and Bagdasaryan. And these are, yeah, we've been interacting quite a lot in the past, so a lot of the ideas that I'm presenting are also kind of shared. And we recently did like a protocol-focused vignettes combining tensor and LIANA. And I will do something, we do something similar in the LIANA Plus manuscript, and I will go back to this hard data set, right? But here we're looking at the dissociated data with, I guess, the additional complexity that we're also taking into account, basically whether ligand receptors and cell types are known to co-localize in the spatial data, right? And this comes from some benchmarks that we did, and if you're interested, I can show some results later. But basically what they showed is that the performance is quite close to random, not random, but any method not spatially informed is, not going to do great if you don't provide some additional input, and this is more or less expected that we have a lot of false positives in these methods. So what we do is that we take the basically the interactions that were important in the different condition related regions from the NMF analysis, and we also do some, again, this like global interactions that we identify with MISI between cell types, and we only focus on cell types that are positively associated in space, right? So basically we constrain any kind of ligand receptor inference from these associated data to ligand receptors that we know co-localize and cell types that co-localize, right? And that's about it. So basically what we do then is we have 29 samples, and we run LIANA on each sample, and then we feed it into basically MOFA, right? So it's like an M-data object that we generate with views, and the views are cell type pairs, and we do basically factorization again. And yeah, what we get is quite nice. That it's unsupervised, right? You are not biasing your analysis to what you want to see, and we see that we have like basically a nice separation between the three groups, like almost like perfect, which is nice to see. And then what is nice about MOFA is that it would give you basically the variance explained per view, and this one basically you can treat as like the how well each cell type pair is like captured in a specific factor, which in turn implies like how kind of important this cell type pair is for like the interactions that separate the samples, right? And we can see, which is quite nice here, is that basically FB stands for fibroblasts, and MIY stands for myeloid. So we see like basically canonical association between fibroblasts and myeloid cells, which is known to basically be a driver of fibrosis in ischemia. And from this, we can kind of have like a hypothesis that what we're looking in the data actually makes a bit more sense. And I think here I simplified a bit more. And in addition to these like factor analysis, we do something quite simple, which is basically we take prior knowledge, we take pseudobulks of cell types, we do differential expression analysis, and then we just join it, right? So I think Boot-Inishnet does something similar, if you're familiar with that. And we get basically deregulated interactions, kind of, right? And what we see is, kind of what we expect already from the spatial data, from the factorization, this dissociated ligand receptor loadings. We see these basically pop up as being significant, and positively enriched in ischemia, right? So that's nice. And I guess the final thing that LIANA does is that it also connects to intercellular signaling. And basically this is done with an intercellular linear programming solution or problem. Basically, we optimize a problem such that we include the least amount of nodes and edges in a way that the network that we get is sign coherent, right? So I think it sounds kind of detailed, but all it does, right, is you get sign consistent paths. So if you have like something that is down or something that is up that reaches something that is down, then the link to like let's say a ligand that is up and a receptor and a TF that is down will be overall negative, right? So it will be decreasing, right? And this is what it does, right? And just to kind of visualize how this looks is that we get those networks which are kind of basically acyclic, right? So they start and the end somewhere. So you can also get the shortest path essentially if you have like one-to-one. And it's quite nice, this network that we get, because we actually capture more or less canonical TGF-beta signaling, which makes sense for the interactions that we're looking at, right? And we see, for example, SMAT1, SMAT3. SMAT3 specifically is like TGF-beta canonical basically regulator, and FOXO3 has also been implicated recently in basically interacting with SMAT3 in TGF-beta signaling in myocardial infarction. And I think this is like more or less a whole framework or like a summary of it, summarized in a couple of applications. And I think here I would thank you basically for the attention. And basically LIANA is the framework that they presented, and I want to say thanks to basically all the scverse people that keep pushing this, because I think it makes life much easier to have like a solid foundation to build stuff on. And I do my best to provide as many vignettes as possible. And I'm happy to discuss and take questions.
Thank you so much. Great presentation. Clap. I mean, it's a pity online, you never hear the clap.
Thanks.
So we're still recording. So if anyone is uncomfortable, they can put everything on the chat and I can read them for you, the questions, or otherwise can please raise hands and then I can actually name the next one. So I can start with the chat. Thanks for the presentation. I'm in a busy room, so I cannot speak. Could you elaborate how you get the spatial factors? Mephisto?
It's the spatial factors. I think in this case, I mean, we can, like the ones that I showed are just NMF. So basically the features that I put into NMF would be spatially weighted, but I don't take space with the factorization. But it's a good question. But I think you could run Mephisto essentially. So you could do LIANA or whatever with Mephisto, or like whether it's spatial or like some kind of like time series. I think there isn't that much data that would fit for this, and yeah, but it would work, right? It's essentially I'm running MOFA in one way or another. You can feed any part of LIANA into MOFA since it's undatas and MUDatas only, so it should be straightforward to do. I don't know if I'm answering the question properly, but yeah.
Yeah, I think so. I mean, they will comment again, I guess. Okay. So, Isaac?
Hey, nice talk. So I have a question about the kind of NMF that was used, which is I feel like there's a ton of papers out there that are like special NMF for single cell data, but I believe you're just using the scikit-learn NMF.
Yeah, it's scikit-learn. I think here I just assume that since the way that these local metrics work, right, is that imagine you have like the SquidPy graph, the spatial graph, and then you just do like a matrix multiplication of X, matrix multiplication of Y, and then you do Pearson or any of the other ones, right? This like in the very simple way, this is how they work. So I assumed like I'm already encoding space, so I just didn't do it, but I think it's fair. I even put it in the discussion that, like depending on the application, one could also do spatial NMF or I think there is a bunch.
Did you end up looking at any of those or like kind of comparing if you got better or worse results?
I haven't. I think my issue with the paper was that I made it maybe a bit too wide. So now I spend like months basically constraining as much as I can. but yeah, I'm super interested to do it though. I think this is definitely an interest of mine too.
Yeah, I do think just basic NMF does seem to work pretty well. So maybe it's a good thing.
Yeah, I think it works like almost like magic, but it's clustering, so it kind of makes sense. Like the clusters that you get otherwise as niches, you get them also as NMF. And the ligand receptors are also like this is also the cell type markers quite often, especially receptors. So almost you can see that there is like a strong association basically between cell type and ligand receptor clustering in a way. So yeah.
Cool, thanks.
Ong?
Hi, it's just I think I have some practical questions to ask since just today and before I was running also NicheNet and today I tried in LIANA. And first question is regarding like the NMF, since it should output like as long as more as almost the input feature number of factors, like factor 1 to n or n minus 1. But when I run it, kind of, if I set like factors equals 8, then I only get 4. And then I test how they relate to my 3 groups. I have 3 groups. Then like only 4 factors could have statistics, t-test statistics. Others are all 0. Then that means like after 4 factors, I just don't have lose the association to the group also. And I don't know how like the parameter choose if sometimes I can only set it to be like 3 factors or 4 factors, but not more.
I think, so if I'm getting it correct, is that you are manually setting the number of factors. And one thing is that I guess I implemented also this procedure that, it's again a heuristic, right? So it's not ideal, and it's kind of dependent on the number of max ranks that you put. But basically, you can do a search of like between 1 and like let's say 50 factors, and it will give you like the optimal result. Maybe this is how you can justify why you limit to 4. So if you put like 1 to 20 or something like this.
one thing I also really want to ask is how if it's better to do more feature selection and sample or know the cell selection before, or if I have a more clear question. For example, I already have a question with regard to which cell type, but not input all the cell type. And also, like, if I would like to, I'm interested in like the VEGF-A receptor complex kind of. If I could only input a subset of the whole single cell data that I have, or it's better that to input everything and let it impute all the interactions there, or
I mean, I think generally you want to, I guess, focus if you have multiple samples on like features that are, shared and variable across. Like, I mean, there is like some rule of thumbs of basically that I remember vaguely that basically you want them to be like some kind of an intersect of the highly variable features so that you get like most of the noise out, right? I think this is also step 2 of, I guess, the ScanPy tutorial. But I think it's also kind of question dependent whether you also do it like for a specific pathway, right? I think it's like if you say that you're specifically looking at like, let's say, soft clusters of like TGF beta, then I think you can probably limit to that. And if you have enough signal, you should get clusters that are meaningful, I guess, or factors.
yeah, I just hope that the subset do not like someone would question that I do not use or I manually choose a certain distribution and not like from the whole data set, or especially when if it will change the results when I only look at 2 cell types mutual interaction instead of like let the model to impute all the potential networks across all the cell types. And because when you move out some node and manually selectively move out some node in the network, I don't know if it's accepted or in a way whether like practically when I write the manuscript in the end, anyone would question that.
I mean, I think it just depends on the question, right? So I mean, I personally, if I'm looking at methods and I see that someone wanted to see factors like or clusters driven by something specific and they did clustering on that, then I mean, sure, but I don't know if anyone else has like an opinion on this, but
Yeah.
Sorry for that. Someone is connected, but I cannot say. Right.
Yes, thanks.
Sorry for
thanks for the questions.
So we have more questions in the chat. So it goes through a bit the conversation on the chat with the first question, but I guess that you can catch it later.
Maybe I can. Should I read them or?
No worries. I guess it's a conversation for the Mephisto house. So, but there is another one. How does LIANA address the sparsity of the spatial data? When I plot individual ligands and receptors expression from Visium data, it often looks very sparse, hard to make out of any pattern. And the second part is, do you think LIANA be applicable to MERSCORE data, more than 400?
I mean, okay, so I guess the first one, one of the things that they do is that I filter out basically any ligand receptors, any of the members of which, including complexes, are not expressed in like, let's say 10% of the spots by default, I think. So I guess this would get rid of like some of the issue with sparsity. Beyond that, I'm not sure how to deal with sparsity in Visium. I think maybe for me, the bigger issue of Visium is like this kind of like soupy effect that it has, but I think usually just filtering out things that are not expressed, for the whole slide probably gets rid of most of the noise. And also, if you focus on things that are variable, I think it's also another way to do it, right? So LIANA should be applicable to MERSCORE data. I mean, I've only looked at it when I'm doing like efficiency or like basically the tests for like when I have more data points. I guess the issue, as you say, is like 400 genes, that you have only 400 genes. I guess not since it's MERSCORE, if I remember correctly, it was like targeted, so many of those will be receptors. Not sure, since they're cell type markers, not sure if many will be ligands, but I mean, essentially you could also, you don't have to apply LIANA on ligand receptors, right? You can also look at like cell types, for example, just as long as you ensure that somehow they're like, yeah, basically the features are in something that would work with LIANA, so like, yeah, somehow normalized or scaled, and then you can look at like bivariate associations between all the 400 genes or the cell types that you have, or that you identify with those genes. So you could do it.
Great. I'm going back again to the, sorry, Tim, I'm so sorry, I just messed it. So he replied that ideally you don't want all latent factors to correlate to your grouping. They should be semi-orthogonal, and that's the answer to Hong's question. Just
Okay.
wanted to read it. And
Thanks.
Emma?
Hi, thanks, Daniel, for the presentation. I had a question on the multi-condition testing part, essentially. So like the hypothesis-driven analysis. And I was wondering if so as far as I understand, you use basically fold changes of the ligand and the receptor, and then you combine them, and then you essentially tell which ligand receptor pairs are more upregulated in a certain condition than another. I wonder if you also tried doing testing at the cell type level. So basically where the levels of expression stay similar, but test whether a specific ligand receptor interaction, for example, the same source cells changes its targets in response to a condition, or do you see what I mean? There's a lot of things that you could change here that you could test on. Also, for example, like if levels of the ligand and not the receptor change. I'm wondering if you've tried a lot of these different options.
I think the way that it works, I mean, I try actually present it like in kind of simplistic, I guess, applications. But the way that, I mean, the function is called df to LR, right? And I guess there is a reason, because like you can have like any kind of data frame that you just join, and LIANA will take care of complexes. But yeah, I guess it doesn't have to be that both also ligand. I don't know if I got the question correct, right? Because I don't know if you don't have to have both ligand and receptors going in the same direction. Yeah, I think it's just in this case I just show those because it's the simplest case. Also, it doesn't have to be PyDesic. It could be like MOFA loadings, for example, or like dialog loadings or whatever you have like prior to that. And yeah, did I answer it? I'm not sure.
Yeah, and one thing was like testing basically for differences in the source and target cells rather than differences in expression of the ligand receptors. I don't even know if that's something you see as happening. So
basically, let's say you have like Milo, and then you do like, you see that something is like a source is enriched, something like this in a specific condition.
I was thinking, I don't know what I was thinking, but I was thinking more a case where the ligand, for example, becomes differentially expressed between two cell types in a specific, like essentially becomes more highly expressed in one cell type in condition A, while it's more highly expressed in another cell type in condition B, and whether you can like use that in some way to, hypothesize that the interacting cells changes rather than the interaction itself. So maybe the cell uses the same type of ligand receptor interaction, but with another cell type. But again, I don't really know what the statistic would be.
I was going to answer in a really like dumb way that like if you have like some data frame with this statistic, then yes.
Yeah.
I don't know if it's a good answer though.
No, I don't know either. I was wondering if, but yeah, it feels like you could also do some sort of think about some sort of testing at the cell type level, because I think some papers that look at disease data sets try to sort of, based on the normal ligand receptor analysis, make some inference about this cell type is in changes who it interacts with, but they're a bit like fishy. So
I mean, you can also, I guess, like run CellPhoneDB and like left join to this data frame, and then I guess it works. I guess like if it's like something to do with like, let's say you have like a time series, and then you have like an emergence of cell types. I guess if you do like this, like other factorization approaches that I presented on the single, like on the dissociated data, and you treat like emerging cell types as biologically relevant, so you basically impute zeros, and then you see like certain interactions pop up, then I guess you can also see it there. I don't know if I'm like even making it like, yeah, I don't know.
Yeah. Well, something to think about.
Yeah.
Great. I also have a question. I mean, it's more practical kind of. So you do the predictions and you do it in different levels, right? You have metabolites and you have can I ask how do you like, because you showed in the presentation, you showed that you benchmark or you validate them biologically, which makes a lot of sense. But is there a more automated way where you can actually benchmark these predictions and say, okay, that's the percentage, that's kind of, I don't know.
Yeah. Benchmark? Sorry, what? Because sorry.
Yeah, no, the
like
predictions kind of the. I'm not sure exactly what's your output. Like I have never used it, right? But so you actually predict the metabolites based on their gene expression, right? Or...
Yeah. I mean, I think I guess you can get stuff like R square, which would be like the variance explained in this specific, like for the metabolite peak by like the other views, right? So in this case, it was cell types and receptors, and I guess this would be kind of like your sanity check metric. I'm thinking also to do like other like metrics of quality, like RMSE, sorry, and yeah, but I think this is more or less, I guess, what you expect to be the one that tells you how well a model performs. But yeah, it also depends like how you model, like what does a good R square. I mean, I guess like for metabolite peaks, they're quite noisy. They're like, I think people that do metabolites say that they're semi-quantitative. So when I saw like a R square of 0.5, I was quite happy. But yeah.
Okay, I see.
Daniel, very nice talk. I have a very kind of stupid question. Actually, when I run the LIANA, which is very nice, I did the pseudo walk and I got the interaction score, which is based on the upregulation, downregulation. I just have a quick question. It's like, I think it's really up to log2 fold change, but like, does this interaction score include the expression level of ligand or receptor as well?
I think in the default in the vignette, what I do is that I rank them by the SWOT statistic, which is the statistic that PyDseq outputs. It would be like the mean SWOT statistic. But I think also I provide like a mean expression basically for each interaction, so you can also look at that. Though I think PyDseq, what is the kind of annoying when you work with, I mean, in this case, right? Not annoying, not the best word to use, but is that they do it like the mean on the counts, right? So it's not normalized. So it's a bit, but what you could do, right, is that you can, I guess, do like a mean on the basically. So use the pseudobulk mean that I think the coupler would actually give you a pseudobulk mean, and you can just append it. I mean, this you can write an issue, and maybe I can think about it a bit more and have it as a default, because it's a good question, and like maybe a limitation of the way that I do it currently. I'm also thinking to maybe like wrap the whole thing into like one function rather than to do it externally. But yeah.
Yeah, thanks a lot.
Great. Are there any more questions either in chat or raise your hand? I mean, I have shared your GitHub as well and your Twitter. I have pinned you everywhere, so you can all find him and talk to him more, I guess. But
yeah, I think issues also works quite well. I mean, I try my best to respond fast, so yeah.
Good. That's good. Zulip and everything for scverse. That's how you will communicate from now on. I think all of you. Okay. Thank you so much then. I mean, I don't see anything else. Let's give 20 minutes back to people's time. And thank you for joining. And thank you so much for the presentation, for being the first one presenting here. It was great. And feel free to contact him, to contact Daniel, and use LIANA. And I see you in 2 weeks in our next community.
Yeah, thanks. Thanks everyone for the questions. Bye.
Thank
you for the attention.
Thank you.